Picture a bustling city teeming with people who are all unique. Now imagine that this city is your body and the people are the cells of your body. We have around 30 trillion cells and they all differ to some extent. For instance, we have red blood cells, which are perfectly suited to transport oxygen. We also have neurons, which act like wires and forward information. Despite their differences, all cells share one common requirement: glucose. This simple molecule is the fuel of our bodies. And if glucose is the fuel, the pancreas is the engine, because it regulates glucose uptake. Even the slightest imbalance in the system can lead to serious conditions like diabetes. This is why pancreatic cells are of utmost interest for analytical chemists at Uppsala University.
In my research group, we are doing research on rat-derived pancreatic cells. If you want to find out more about these cells, let's follow me to the lab. This is our cell lab. We do all the cell culture work here because it has a special sterile environment and it's very important for the cells.
2 hours later.
In the cell lab, we need to put on protective gear, both to protect us and the cells. The cells are cultured at 37 degrees in an incubator. You can't see them by the naked eye, but with a microscope you can make sure that they are alive and thriving. Since the cells are growing attached to the wall of the flask, first we need to detach them with a special enzyme cocktail. Afterwards, they are resuspended and transferred to our fancy machine, Selena Gomez. The machine detects single cells and isolates them to predefined spots on a glass slide. In the right panel, you can see the single cells captured in the capillary that is moving very fast to isolate a large number of cells in a short time. Once the cells are sorted, we place the glass slide in front of a machine called mass spectrometer. Here, we can detect different molecules that the cells are made of. This happens with the help of two thin capillaries. Whenever these capillaries touch a cell, we can see a signal increase.
Last but not least, we need to handle the data. This requires a lot of programming, which helps us to distinguish healthy cells from diabetic cells.